So we just learned about the structure of DNA and how that was discovered, right, by Watson and Crick and Hershey and Chase, Rosalind Franklin, all of those famous people. And so Watson and Crick discovered the double helix. So there's two strands, they run anti-parallel to each other, and that they are in this helical spiral staircase form. That was their discovery. They also realized that the base pairs line up together, just like we saw in a recent slide, maybe. Base pairs line up together like this, guanine with cytosine and adenine with thymine. And we can see here that that's how it always is. So if you have a GCG on this side, it's always going to be CGC on the other side. And so basically their discovery kind of led to the idea that DNA contains the instructions for itself to be copied, because one half of a DNA molecule will always be able to tell what the other half should be, always. And so there was actually a section from their paper, does it say it in here, where that they talked about this. I'm going to read it to you. It says, now our model for deoxyribonucleic acid, DNA, is in effect a pair of templates, each of which is complementary to each other. So we have these two strands, and they fit each other perfectly. They're not identical, they're not mirror images, but they together make a perfect pair. They're complementary. That's what that means. And continuing, we imagine that prior to duplication, the hydrogen bonds are broken and the two chains unwind and separate. Each chain then acts as a template for the formation onto itself of a new companion chain, so that eventually we shall have two pairs of chains where we only had one before. Moreover, the sequence of the base pairs will have been duplicated exactly. So Watson and Crick were able to come up with this, and they were totally right. So they're complementary for each other, so if we break them apart, they each are a template, a format, rules to follow to make the other half again. And so basically, this section is now about DNA replication. So we have our parent molecule right here. A matching with T, C matching with G, T matching with A, et cetera, for 6 billion base pairs, right? And so what happens is these two strands get separated. They get pulled apart by a mechanism we'll talk about a little bit later, but they get separated, and now we have two single strands. And this only has half the information, but it also contains the information for the other side. So here, yeah, this is an A, and it was an A right there, but it also says A only ever matches with T. So it's perfect because on the other side of this, there must be a T. So this side has the instructions for the other side to be made all over again, because this must have a T right here. This must have a G right here, and so on and so forth for 6 billion base pairs. So that's the incredible thing about this, is it contains its own instructions to replicate itself. So what happens is they get separated. Now we have two templates. So this one has the instructions to make the other half, and this one has the instructions to make the other half that go right here. So then what happens is we get two daughter molecules where, by a mechanism we're going to talk about, these other base pairs get brought in and match up perfectly. So we know that if there's an A right here, there's got to be a T. We know that if there's a C right here, there's got to be or a G right here, there's got to be a C. So we can make a perfect copy of what we already had. If you look at this, it's exactly the same as this. Exactly. If you look at this new molecule right here, it's exactly the same as this one. Okay, so they just made perfect copies of themselves because they contained their own instructions for doing so. And this is called the semiconservative model. So Watson and Crick thought of this, like I just read to you, and basically it says that when a double helix replicates, they separate, and each daughter strand, or each parent strand, contains the instructions to make a complement. And so each daughter molecule is half new, half old. And so that's what semi-conservative means, semi meaning half, and half of the parent molecule is conserved into the new strand, and that's where that comes from. This is the two other models that were thought of were the conservative model, where the entire parent strand is conserved. So basically, they make instructions for, they get copied and a new strand gets made, and they go back, they connect again, the parent strands. So the entire parent molecule is conserved, or the dispersive model, where pieces of the parent molecule end up in all 4 of the daughter strands. So here's a picture of that. The conservative model, nope, The conservative model says that the parent strand stays together forever, so the parent strand is a darker one. So the first replication, we make a new copy, and the new copy is entirely new, and the old copy is entirely old, and so on and so forth. So by the second replication, the parent molecule is untouched, it's still the same. Whereas the semiconservative molecule, or model, shows that one strand of each goes into one of the daughter molecules. So here there's one new strand and one old strand. Here there's one new strand and one old strand. And so if we duplicate again, the parent strands are still staying intact, but they're just ending up in new molecules with new pairs. And the dispersive model is shown right here that some ends up, some new strands, some old in here, some new and some old here, et cetera, and keeps kind of getting broken up. There wasn't a lot of proof for Model A or Model C, or really anyone having any idea how that worked, but they were just ideas that couldn't be thrown out until they were proven to not be true. And so then came the Michelson install experiment, which you guys are familiar with. Check out that other video that I posted if you need help with that. And Michelson install basically confirmed that, they confirmed the semi-conservative model. Okay, so let's get to DNA replication. So we've been talking about different models for it, and so now we know exactly what happens. And it's a big, long, hairy, detailed process that you guys have to know, but it's very straightforward, but there's a lot of little details. So basically it happens really quickly. A bacteria can copy all its genes in about an hour. Now we have about 1,000 times as many genes, or 1,000 times as many base pairs as a bacteria, but we can still copy our whole genome in a couple of hours, which is pretty incredible. And the amount of mistakes are almost nothing. About 1 in 10 billion base pairs is wrong. So that's pretty accurate. Oh, and there's a lot of enzymes and a lot of proteins. It's a lot of them. So basically what happens is the DNA has to start somewhere, the replication, and remember that we need the parent strands to separate. That way we can get at the template that they form. So that happens with an origin of replication. And an origin of replication, let's look at the bacterial chromosome first because it's a little bit smaller and easier. Basically, here's our double-stranded bacterial chromosome. Remember, their chromosomes are circular. So here's our origin of replication. The DNA strands get separated at the origin of replication, and then we have what's called a replication bubble, because there's just like a little bubble between the DNA. So that's where the DNA replication is going to start. And basically, DNA replication happens both ways. So it's going to go this way, and it's going to go this way around the whole bacterial chromosome. Keeps going further and further out. See, it just keeps going, keeps going. And you can see that the strands don't unwind until DNA replication gets to it. And then eventually, we have 2 new molecules. So it just went around in a circle both ways. DNA replication goes in both directions until we had two new daughter DNA molecules. And you can see one strand is old and one strand is new on both of them due to the semi-conservative model. That's bacterial chromosome. We use the same methods, but theirs is much simpler because they have one circle and far fewer base pairs than we do. Now, eukaryotic chromosomes are much longer. We have more of them. We have 46 inside of our cells, and they're linear. So it's we can't start in one spot and expect to get back to the other spot because they're linear. But aside from that, it starts in the same way. So what happens is there's an origin of replication. Now, instead of just one, like on a bacterial chromosome, there are hundreds or thousands of origins of replication per chromosome. So there's one right here, there's one right here, there's one right here, et cetera, et cetera. And the same thing happens. The strands get separated, they form 2 templates, and then replication occurs, and we have a replication bubble. Here's one bubble, here's one bubble, here's one bubble. One replication bubble per origin of replication. And then replication happens in both directions again. So it starts to the right and to the left, to the right and to the left, to the left and to the right. And it just keeps going and going and going. The replication bubble gets bigger and bigger and bigger until eventually the replication bubbles meet each other and we're done. And we have 2 daughter DNA molecules. Remember, semi-conservative model, 1 old strand, 1 new strand every time. So what these pictures are showing you are the replication bubbles. So this is the old strand right here, and the replication bubble is getting bigger and bigger and moving outwards, like that. And here is a bacterial chromosome, so you can see here's, I'm sorry, a eukaryotic chromosome. Here's one replication bubble, here's another, here's another. And replication is going in both ways. And these areas where the DNA strand is being pulled apart and the replication is happening this way, is called the replication fork. So at the end of each replication bubble is the replication fork, which is a Y-shaped region where new DNA strands are elongating. So basically, it's just going like this, and the whole thing is replication fork. So on bacterial chromosomes, there's two replication forks, one at the either end of the replication bubble. Replication fork right here, replication fork right here. On eukaryotic chromosomes, there's lots of replication forks. One right here, 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 one right here. Basically, it's where the parent DNA strands are still attached to each other, but they're opening bit by bit to allow replication to happen. Okay, so we have to separate these strands somehow. Remember, they're held together by hydrogen bonds. So it is helicase that goes along and undoes these hydrogen bonds and opens up the two strands of the parent molecule so that replication can happen at the replication fork. So helicase is kind of going along the replication fork and opening up the process as we go. Do you have a picture? Yes. So here's helicase. This is the replication fork where the two parent strands are no longer attached but are still attached right here. And then the helicase is just working its way down the molecule, separating these base pairs. So this is the replication bubble. This is the replication fork right here. And here's helicase working way down there to open it up. That's one of the important enzymes or proteins. Another one are called single-stranded binding proteins. And you can see those gray guys right here and right here. And the purpose of single-stranded binding proteins is to make sure that the two parent DNA strands that we just separated don't automatically go back together. Because they want to be together, they spend most of their time together, but in this case we're trying to keep them apart, that way we can use them as a template to make new strands. So what the single-stranded binding proteins do is they just keep the two strands separated. They keep them single-stranded instead of double-stranded. They stabilize the single-stranded DNA, is what this is. And then we have topoisomerases. What that does is keeps the whole DNA molecule from getting tangled. Because basically, remember this thing's really twisted. Double helix the whole way up, and suddenly we're just pulling it apart. And that could cause some serious twisting to happen. So what the topoisomerases do, they just prevent that from happening. They relieve the strain that's put on the DNA molecule when we're pulling apart the two pieces by breaking, swiveling, and rejoining the DNA strands. So for example, let's say I had a purple strand and a white strand of DNA, and they're all wound together because that's the structure of DNA. Wound, wound, wound. And then I pick a replication, an origin of replication. Let's say this is my origin of replication right here. And then I form a replication bubble, and then I just keep going. The replication forks keep going. My pipe cleaners are getting all tangled and twisted. I mean, they're pretty short, so they came right apart. But the topoisomerases would prevent that from happening. They just keep things from getting tangled, basically. Okay, so now we've separated the DNA strands, so now we've separated them, so now we want to make the DNA. We want to make a copy of it. I'll talk about that in the next video.